time, but we want to prove it with the time.
Okay, thank you for your answers. Rudy, do you have another questions?
So I noticed on the document, on the part of bioreactor scaling, that you have some, looks like a Monod equation, as well as some continuous and plug flow equations for space-time. Can you elaborate on what is the purpose of those equations and how would you apply it to make the plasmids that you're interested in?
Joe Sebastian, please.
Yeah, we propose use these two equations of space-time using Monod, and because part of iGEM is to scale, and we wanted that this project, if we continue this project, and we can scale up this project, and we have to determine which reactor will be convenient for us for this specific reaction. That we will be creating biomass with the transformation, yeah, from the with E. coli, and yeah, I wanted to see how will be better for us if we use a plug flow or a mixed tank reactor, and how this will affect the production of plasmids.
Okay, now we have reached the 10 minutes of questions, and I saw that Judge Niskak had a question about the project.
Yeah, thank you very much. I just want to ask a very quick question. I want to thank the team for doing a good job. Can you tell me what informed your choice of the design binary plasmid as a vector for your project, given that there are other designs you would have used or adopted? What informed your choice of that particular design for your project?
Go, Luigi.
Well, we decided to use this binary vector system. First, because we were doing research and we saw that not most, but many transformations had been used with this system in plants in order to fight phytopathogenic fungi. But well, now we can use just one plasmid, but we decided to keep it so that we can then make a permanent transformation if the farmer wants it. So we wanted to keep this binary plasmid and put all our cassettes within the right and the left border. So we have the machinery in order to make the transformation to the plant. And in order to make that, we would have to add the Ver helper plasmid, which contains the virulence genes, in order to make, to take the information that is within the right and left borders of our plasmid and introduce it into the plant's genome so that the expression is permanent. It introduces into the genome, but it is thought for future developments of the product if the farmers want a permanent transformation. So originally we want just to make this transitory transformation, but we kept this this system thinking of a permanent transformation, to make them a permanent transformation.
Oh, that's great.
Okay, Judge Rudy, you had another question? I saw your hand.
Yeah, it's fine. We're over time, so I think it's fine. Thank you, though.
No, we also have 5 minutes left for more questions.
Oh, we do? Okay,
yeah.
Great. In that case, just wanted to see that, when you're scaling it up a bioreactor, right? You mentioned that there's two types, CSTRs or continuous stirred bioreactors versus plug flow. So based on the equations and your experiments, or not experiments, but potential kind of hypotheticals, which is the bioreactor that makes the most sense to use to produce plasmids, and why?
We think that the CSTR reactor will be more convenient for us because, like we know, when the E. coli is growing, it's going to produce CO2, and we are going to have a gas flow, and in a plug flow reactor, the liquid medium and the gas flow, like the mix, going to have and is going to have more pressure in our system. So we think it might be, like, theoretically will be more convenient to have a CSTR reactor.
And just, you know, I know it's an odd question, but that's what people do to make plasmids. So good answer.
Thank you. Judge Andres, do you have a question for the team?
Yes, I do. Hi, everyone. First, congratulations, guys. Your project is really, really interesting. It's obvious you spent a lot of time on it, so great work. And I would like to ask you guys your next steps, basically. So after all of this is done, what are you going to do next?
Go, Sebastian.
Firstly, we are going to start with the like we have talked with our director of the career of biotechnology, and he wants to to study more the carbon dots. So that's what we are going to do first, to produce these carbon dots and to study the diameter that of the carbon dot that we produce, because we are doing from coffee products. And then when we know more about our carbon dot, we want to study more with the interaction of the positive charge of the carbon dot and the negative charge of the plasmid. How is that going to work? And with that, we are going to do some work with bioinformatics, because this is also like the molecular interactions. We have to study more how it's going to adapt the plasmid to the carbon dot, and then we are going to do all the experiments with the plants. Like, that's fully the order.
Awesome. Could I follow up? So you're going to evaluate the health risks as well of the carbon dots. I had doubts about that. If you had researched if they have any effects on humans. And I also wanted to ask you, because hopefully you get to a point where you actually make this product and you can I don't know, start your own company or sell it, right? That would be a long-term goal, I imagine. So my question would be, and this is what maybe some consumers would ask you, what makes your product better than some other traditional methods that could also solve this problem, right?
Yeah, regarding the biosafety of the carbon dots, the carbon dots are referred as low or very low toxic. That's non-toxic because it's the nucleus of the carbon dot is carbon, so it's not it's not going to affect the plant directly. And what was the other question?
So let's imagine you carry out the entire process. You have your product now, your carbon dots, your fungicide. What would make that better than a traditional method, so you can actually sell this to consumers?
Like my teammate Luis said, like the easy form, the easy way to use our product, that is via spray. there is something like, if you compare using a spray and using a gene gun, or I don't know, another transfection technique, spray is easier to understand. Like, the instruction is apply, like we read papers that they use GFP plasmids, and the way they use it was spraying twice a week for a month, and it was over. So this is, we believe that carbon dots are have a very benefits in, like, to use it with not with a lot of people.
All right, thank you. Great job.
Well, just a final thought, Licky, because we're already run out of time. Just be really quick, please.
I just wanted to add that using the carbons in this method, it's better than using the traditional pesticides and chemicals that can pollute all the environment. And yeah, the application is easier and safer than using the traditional pesticides.
Well, thank you so much for your answers, for your project. I saw it and it's really awesome. And it's amazing that you had like this new experience in iGEM. So the best of lucks, and say goodbye to your judges.
Congratulations, team.
Bye, thank you.
Bye.
Thank you.
It was very nice to meet you all. Have a
Bye.
nice day.
Thank you. Bye.
Okay, we're just going to wait for the next team. And now I forgot to tell you, we were able to fix the live stream, so we are currently live streaming, if you didn't realize in the middle of the session. Just, we're going to wait for the next one. Daniel, are you in there? Let me see.
Tenemos que cambiarnos.
Hey everyone, welcome to your Q&A session. Please remember to turn on your cameras, and also please rename to know that you're students from the team, please.
Primero el nombre, no de equipo. Sí. Ah, puta madre, es que quiero renombrar, pero no.
And this is a reminder that you're being live streaming, so please be careful with your mics. Thank you.
Thank you.
Okay, before we start, I think we have time for a small introduction for the judges. If Beatriz, would you like to start?
Yes, welcome team to your Q&A session. We're all very excited to have you here. Congratulations on your hard work so far. Do you want me to go to questions, or do you want me to introduce myself? I didn't get
No,
it.
introduce yourself, please. Where are you from? Your
Yeah, sure.
institution?
So my name is Beatriz. I'm from Brazil originally. I did iGEM back in 2014 when I was an undergraduate student, and then back in grad school in 2019. It was a super rewarding experience, and today I work as an organism engineer at Ginkgo Bioworks. So right now I'm in Boston.
Nice to meet you.
Thank you, Beatriz. Andres, would you like to follow up?
Hi, everyone. I'm Andres Orozco. I'm from Guadalajara, Jalisco, here in Mexico. So I'm familiar with your university, definitely, and I'm a high school science teacher. I'm look forward to hearing from you guys.
Thank you. And then we can go with Farouk.
Hello, everybody. My name is Farouk Abdul. Actually, I'm from Egypt, based in Cairo. I was part in iGEM in 2019 as a team member, and this year I'm a mentor and a judge. Nice to meet you all.
Thank you. I think I left for a minute, but now I'm back. And the next judge is Sikak. I saw you. Yeah.
Hello, everyone. I'm Dr. Sikak Abasetum. I'm a senior lecturer in the University of Uyo, Nigeria. I was iGEM judge last year, so this is my second time, as this year is making my second time as a judge for iGEM. I'm pleased to meet all of you.
Thank you. And last but not least, we had Rudy Han.
Hi. So apologies, my son is sick, so he needed to get some help. But anyhow, my name is Rudy. I'm currently located in Los Angeles. I work as a principal engineer at the UNIT. My background is in cell culture and fermentation, so that's why, even in the questions, I kind of focus on that part of it. Unfortunately, when I was an undergrad, iGEM got started, so I was not part of any team. So this is my first involvement. Super excited to help out and see what the teams are up to down in Latin America.
Well, thank you for your presentations. And right now we can start with the Q&A session. This is going to work this way. Please raise your hand, judges, to do your questions. And one of the team members opens their mic to answer. And also if you have like another thought, you can also contribute with your answer. So which one of the judges would love to start? Go, Beatriz, please.
I can start. So on your project, you want to use Bacillus subtilis to produce biosurfactants to get the plant in an alert state. Has this been done before? Does this affect the plant growth or the fruit growth at all?
Can you repeat that last thing you said?
So if leaving the plant continuously on an alert state, if this has been done before, and if so, if it was done before, did it affect the plant growth or the fruit production and the fruit growth?
Okay, Alejandro, you can answer.
Actually, most of our parts of our genetic circuit were taken from the iGEM database. So some teams from years ago of iGEM did like some kind of similar things, but it wasn't like did before. It's the first time it's made. That's what I want to say. So about if it affects the growth, yeah, and it also affects the quality of the fruit, and it's also part of the objective of our project.
Okay, I think we have another answer.
Yeah, first of all, sorry, I think my name is wrong for some reason. I'm Adolfo, by the way. But just completing the answer of Alejandro, yes, it has been done, but not by an iGEM team, at least with the biosurfactant that we are proposing. In this case, it's surfacting, and it affects in a positive way. It also not only improves the immune system of the plant, it also improves the quality of the plant itself and the the fruit, the berry, and yeah.
Okay, please go, Farouk, with your question.
Yeah, can you please talk a little bit about your biosafety regulations that you use to produce your project?
Regarding biosafety regulations, we consider the Mexican regulations and also certain protocols that are useful. In the case of the Mexican regulation, the specific name is as follows. It's a norm from Semarnat, which controls these aspects, and they suggest a comprehensive method to evaluate risks. So you need to provide the parts that you're going to be working with, and also you need to provide everything regarding, yes, the risk analysis, either for the project itself and for the environment. So we consider specifically the NOM 002 SAG-Bio Semarnat 2017, and that's required to work with the genetically modified organisms. And regarding the specifically biosafety part, we consider the classification of our organism and also the possible effects that it could have in the environment. So considering the Semarnat and the classification, we determine that it is not dangerous to the environment and to human beings.
Okay, actually what I mean, how does this regulation affected your design in the project? How you are going to implement those regulations side designing the circuit?
For the design of a circuit, actually, regarding, now I understand what you're looking for. Regarding biosafety, in the design of the project, we researched the rates of production of the chemicals, of the objective chemicals that we're expecting. In that case, surfactant and antifungals. So by researching the minimum lethal concentration, we can assure that through the minimum lethal concentration of any antifungal, it is indeed possible to have a safe project, because we won't be stressing the plant and we won't be releasing any more antifungal or excessive antifungal substances into the environment. And regarding surfactant, due to it being a nutritionist chemicals for the plant, we determined that in that case, it is not risky, and we revised every part to determine what it would produce and the effects that it could have in the environment. And specifically, the part that produces the antifungals was the one that we researched the most, because that's the one that could have any detrimental effects. And by using the minimal lethal concentrations that we research, we avoid those external effects that could affect the environment and other human beings.
Okay, thank you.
Okay, Rudy, you had a question, please.
Yes, my question is, the background of vaccines is that, we don't really understand immune systems that well. It is orchestra of a lot of different cells, so it is difficult to understand if something works or not. So with that as a background, if this vaccine would not work, what kind of things would you do to improve and make sure that the vaccine works in the next iteration?
Who would like to take the answer? Okay.
Thank you for the question. Yeah, there is in literature, there is presented a lot of different elicitants that are these molecules that improve or helps to maintain the plant in an active immune state. So if we use the surfactant that we want to use this metabolite because it's present in Bacillus subtilis, that it's naturally used as biocontrol agent, we will try, if the surfactant doesn't work, we will try another elicitant already present maybe in Bacillus subtilis, or try maybe another chassis, but only by testing in lab work, we could reassure that the product that we're proposing, we can, well, make a fit of vaccine.
Thank you.
Okay, we already passed the 10 minutes of questions, and the next question is going to be for the Judge Sikak.
Okay, thank you very much. I want to thank the team for trying to solve the problem of pest infestation in berries by using phyto vaccine that produces antifungal proteins. I just have two quick questions for you. Your work was very interesting. First, how will you sustain the production of these phyto vaccines?
Go ahead, Alpo.
Yeah, for the vaccine production in our organism, Bacillus subtilis already produce surfactant, but not in the concentrations that we want to maintain the plant in an active state, immune state. So our first part of our genetic circuit is going to overproduce an enzyme, the one that is called VU1, I'm sorry, which is proven in literature that it overexpresses this enzyme that produce then as the surfactant. The reason is that it impacts in the leucine metabolism, that it further produces more surfactant that could be excreted to the media since it's a bacillus.
Okay, good. Another question that I want to ask very briefly is, what makes your product distinct from other products in the markets?
Do you have more information related?
Yeah, I'm sorry. Our solution is different because it has two active ways to solution this problem. That is the biosurfactant, and in the same time, when it detects some kind of fungi, it also attacks the fungi. So it's like 2-in-1.
Awesome. Thank you very much.
Okay, that's a good answer. Please go, Judge Andres.
Hi, everyone. First, congratulations on your work. I thought it was great that I could see you spent a lot of time interviewing workers where they were growing the berries. So that's good because we can learn a lot from them that they are there, no? They're on the site. And I would like to just ask you what your next steps are in your project.
Sorry, regarding lab work, human practices, overall.
Oh, well, yeah, you could basically summarize all of it for me. Where are you currently? Because I think you still have some testing to do from what I gathered. So how much testing have you done? What testing are you going to do next? And how would you get to your final product?
Right, about the testing protocols and all of that, I am not strictly qualified to answer that. I focus more on the human practices part. For at least that section of the project, there's a lot of answers that we haven't answered yet. For example, in what we've discovered was that the berry industry was divided specifically in people who are associated with corporations, exporters and markets, and people who are more independent, like they have their fields and they work them on their own, which are actually very scarce. There are not a lot of people who do that. So mainly that would be the first thing that we do next year. This year we only had the chance to talk with people who already have these associations with corporations and all of that, but we are still interested in knowing what happens with the people who are more independent, who are more state-of-the-art, more craft, organic. And we also had the chance to talk with an engineer. His name was Giovanni, and he talked to us about there are some government projects that are starting to rise to the technification of the crops, sorry. And that technification of the crops for these independent people. So that would be also a next step for the human practices approaching to these government installations to ask what are they doing, and if we could implement our project in that technification, if they see it viable, etc., etc.
Okay, please go, Adolfo, and after that, Claudia, please.
I think that Claudia could be better first than me because she's more regarding to human practices, and I'm going to talk about more of the build and test in lab reward wise.
Well, I was going to say that we're also working in the financial aspect, all of the financing, the business model design. We have a lot of work to do to be able to finance our project and to be able to run the appropriate tests in lab. And you can go ahead and answer about the lab work.
Thank you, Claudia. Well, after this validation that is going to be made by human practices with our stakeholders, we could start going and test in the lab what we propose in our genetic circuit by first making a transformation in a model organism such as E. coli. And if we see that all the proteins, all the everything that we propose works just as fine after measurement and whatever, then we can go ahead and build it and test in our chassis in Bacillus subtilis to not only evaluate if the enzyme that we are proposing is produced, but if the surfactant increases the concentration as in the normal level. And after that, we will have our minimum viable product.
Okay, Sophia, would you like to go with final thoughts?
Sure. Also, in the communication area, we're going to continue to share information about science, synthetic biology on our social media. We're also have been creating these videos of each crop visit we have done. So we're going to continue with all this to make our project understandable for all the people who reach to our social media. So that's from communication area. We're going to share this information and all the project updates we have.
Okay, one of the judges have a final question. No? Okay. Well, I think we're just in time. And with that, I would love to thank you. I really love your project and you did a really nice job. So thank you all. And we'll see you later. Thank you for being here.
Thank you for asking.
Thank you so much.
For all your questions.
Thank you.
Congratulations, team.
Bye. Have a great day.
Thank you. Goodbye. Nice weekend.
Thank you. Goodbye.
Okay, with this, we're going to wait for the next team because at some point we get the time confusion. But I hope they are coming soon. Well, besides the technical issues, how are you feeling?
Good. I think all the projects are really, really great. So that's why I'm so curious to see what they're going to do next, because what they've done so far is already outstanding. So I hope they actually continue with it, no?
Yes, totally. And it's like a nice perspective to start thinking, what can we do next? And to start talking about all these new approaches that we can have and new applications. Okay, I see that the team start coming.
Yes, hi.
Hi, Charo. I'm sorry for the time confusion. We got a small delay at the beginning, so that's why we got all this moved.
Yes,
basically.
yes, don't worry, don't worry. We were also kind of confused on what was going on. Thank you.
Yeah. Okay, we are going to wait just a couple of minutes so everyone is on board. Okay, I think they are all coming. Just a quick reminder, after this session, we are going to have a break. So I think we can catch up with the next teams on time.
Some of my mates are telling me that they can come in because it's like the host had eliminated them from the session.
Oh my God, what happened? I don't know what was going on. Let me see with Daniel if he can help us. Daniel, are you in here? Give me a second and let me see if I can fix that.
Yes, thank you.
Hola, hola.
Hola Daniel, tenemos un detalle. Unos integrantes no pueden entrar a la sesión.
Del equipo que está en turno, que se llama Firefighters.
Ajá, es que hace rato entramos, pero no era nuestro tiempo.
Ah,
Entonces,
¿lo sacaron?
y como nos sacaron, ya no pueden entrar. Bueno, les aparece que como los eliminaron por el anfitrión, ya no pueden meterse.
Sí. Problemas. Déjenme cierro porque tengo. Estoy revisando cómo hacerle. Creo que lo que vamos a tener que hacer es que se conecten de otro dispositivo porque su dispositivo quedó bloqueado. Entonces vamos a verlo con tiempo, con calma, no se preocupen, empezamos su sesión en cuanto estén listos.
Perfecto, Daniel, muchas gracias.
Peor escenario posible, si no se pueden conectar, yo creo que iniciaríamos una nueva sesión con ustedes porque, bueno, fue cuestión del delay. No problemas suyo, no se preocupen. Entonces, vamos a dar les gusta 3 minutos, 5 minutos para intentar, si pueden. Si no, porfa, repórtenos y los pasamos al final para que no haya problema. ¿Vale? Farouk, sorry for the Spanish. We're having problems letting people in.
Yeah, it's totally fine. It's okay.
So if everything goes well, we can start the session as soon as possible. If not, please let us know. The team will let us know if we will need to start another session, and probably just to respect the schedule, we will have your session right at the end of the other session, right? So sorry about that. I will be here, but I need to go to another room to solve other issues. Wait a second.
Yes, thank you.
Don't worry, I think they are all starting to get in. How many of your team members are going to be in here?
I believe we are like 12 or 13.
Okay.
Sure.
Yes.
Also, please ask them to rename themselves, because if they only have the name, I don't know if they are in the team. So that's also another issue. I also think what they can do is to log in with your account since you're already here. I think they can pass the credentials so they can also join from a different device.
Yeah, we are still missing 3 members of our team. They still have problems even if they try log in with another device. I don't know if we can change maybe the hour, as Daniel said.
Yeah, don't worry at all. In this case, as I was telling you, probably you can try with your account back in their device if you are confident enough, and probably that can work.
So what happens?
They can still log in, so the best is to move the session.
So we should start the break. So judges who are there? Rodolfo, Andres, Farouk, Sikak. Do you have any issues regarding the time? So we can start this session 1 hour from now?
I'm fine. So like 1 hour exactly, or like, like 15 minutes from now, so that it's on the hour.
So the idea is to start the next session as scheduled, Team 4 and Team 5 as scheduled. And right after we finish with Team 5, that I think this is Group 3, I don't remember which team is, we open another Zoom room. So the ones that were banned can join. So if you have no problem with that, we should reschedule Team Firefighters to 1 o'clock Mexico time, 3 o'clock Boston, New York time. Is that okay with you?
Yeah, it's fine by me, but time in Egypt now is 8 p.m. Can you just send me the times in private so I can adjust it? Yeah.
Sure. So we will have a break now, 10-minute break. Go and take some rest, refill your coffees. And sorry.
Thank you, Daniel.
Sorry,
thank you, everybody.
Firefighters, please join again exactly in 50 minutes. I will let you know via Discord the correct Zoom.
Okay, thank you.
Thank you.
Thank you.
Okay, then I'll see you in 10 minutes then. Thank you, and I'm sorry for all the inconveniences. Hi, team. We're going to start in a few minutes. Please, this is a reminder of keeping your mic off and also your cameras on, and rename yourself with your team's name, please.
Hello, good morning.
Hello.
El equipo One, ¿están listos?
Yes. Ready for here.
So as soon as the judges are also ready, we can start. We have 1 minute left. So please just wait until all the judges are ready to go. Well, wish you the best luck and success, Team One.
Thank you very much.
Thank you.
Thank you.
I will be here if you need something. Well, kind of here because I'm on other stream right now. So if you need something, Sophia, please let me know in the chat.
Thank you. I hope that we are not going to have any other issue. Okay, as Daniel said, we're just going to wait for the rest of the judges, and then we can start your Q&A session. The judges already know this. They raise their hand to answer the questions, and then you can answer it opening the mic. And also, if you have like another thought related to the answer, please raise your hand so we can give you the word. Yes?
Yes, thank you.
Okay, Judge Sikag and Judge Andres, let me know if you're in here so we can start. Okay, we're only waiting for Judge Sikag, please. Okay, well, before we start, you're going to have 20 minutes to answer Q&A session. And I'm going to let you know once we already have 10 minutes into the session and once we have 5 minutes left. So I think, so we don't get like any more delay, we can start if everyone is okay with that. So welcome to this session, and which one of the judges would love to start? Okay, please go, Farouk.
So, yeah. Actually, great job, a great effort. I really liked it. I was just curious how you are going to target a specific insect, like affecting other insects, and this might affect pollination and other stuff in the ecosystem. How we are going to target the specific insect that's causing.
Sorry, we didn't hear the question. It was a lot of noise.
Yeah,
the audio.
It's okay. It can be. Yeah, I was asking about how are you going to consider that your project is going to target the specific insect or the specific pest, not affecting other pests or not affecting other insects inside the environment or in the ecosystem. Is it clear?
Okay, let's go, Adrian.
Yes, that's one of the reasons why we decided to use spider venom peptides, as these are highly specific molecules we investigated a lot of options and filter out based on if they affect other insects that are beneficial for the environment, if they affect mammals, and most importantly, humans, and choose two peptides that are specific to Coleoptera insects. And even though they also target other orders of insects, there are also pests from these categories, so it wouldn't be as problematic.
Okay, great, thank you.
Okay, please go, Beatrice.
Hello, team. Are the peptides you chose allergenic? Could someone have an allergic reaction from eating nopal rather than that isn't allergic to nopal, but is allergic to spiders, for example?
No, please.
Okay. We didn't test this yet because all we did was in silico job, but thanks to our bibliographical research, it is not likely to happen. There might be a probability, but it's really, really, really low probability.
Okay, Rudy, do you have a question for the team?
Yeah, so I think if I recall correctly, the way that this is expressed is that you're trying to do the two peptides and you're repressing them and expressing them with different sugars. When you grow this in a bioreactor, if you have a culture of two different organisms and you're using sugars to repress an organism, but the other organism is going to express it, like what's going to happen to the repressed organism with that sugar?
Okay, go, Grecia.
Yes, we had we explored two different possibilities. In the first one, we wanted to express like the same circuit, but with different venom peptides in different strains. So if we and with that we will express it using methanol as an inducer, and in that case we will use different bioreactors. And for the second option, we're going to express both peptides in the same strain. But the thing is that, with one we will have the expression of one peptide, and with the other carbon source, we will have the expression of the other one. However, our project was more focused about the first option because we're still the second option is only a perspective that we would like to explore in the future.
Thank you. Emmanuel, do you have a follow-up answer?
Yeah, I would like to share my screen. Here we have an overview of the production process in bioreactors. We have a growth phase with glycerol that works as a repressor of our genetic circuit. In this option, we are going to produce our peptides in different strains. And then we have a transition phase where we are going to start adding the inductor that is going to be methanol. And finally, we have a production phase that is methanol feed batch phase. And then in that phase, we are going to start the production of our peptides.
Got it, perfect. So just if you don't mind, follow-up question is, methanol can be toxic at high concentrations. How would you avoid the methanol from being a toxic compound in your expression system?
Okay, the feed batch will allow us to feed the culture with a specific concentration of methanols. We are going to determine which concentration we will use, and it will not have this toxic effect on our strain.
Okay, thank you for your answers. Another question for the judges? From the judges.
Okay.
Yes, go Beatrice.
Go ahead.
How did you choose your fusion protein? How did you get to lecithin? How did you select for that?
Okay, this was an idea from one of our advisors, because these peptides, these insecticidal peptides, are not meant for oral consumption. In nature, they are injected by the spider into the prey, causing an immediate biodisponibility. But that's not the case for our product. Here in Spidicide, what we are doing is spraying the biopesticide into the nopal, and the goal is for the insect to consume it, to eat it. And even though these peptides are pretty stable to environmental conditions, they are not strong enough to survive the digestive system of the insect. And even less, they don't have a mechanism to be absorbed into the hemolymph of the insect and reach the central nervous system, that is where they have the toxic effect. Therefore, we needed to find a solution to this, and that's how we decided to use a lectin. The lectin is a protein used by plants as a defense mechanism to do something similar than us, to fuse it with a toxin protein with a toxic effect to defend against insect attacks. So what we did was use lectin that increases the stability to sustain the pH of the stomach and allows the absorption in the hemolymph of the insect.
Okay, thank you. Sikak, you had a question for the team?
Yeah, thank you very much. I just have one question for this team. I want to commend the team for doing a very nice work. The job is, when I was reading through their project, it was very interesting. The production of Spidicide as alternative in the management of cactus weevil pest. I don't know. I want to find out from the team what market strategies they intend to adopt in order to introduce these products in the markets to customers. What marketing strategies would you adopt in order to introduce this product to customers?
Okay, go Andre.
Okay, so for the marketing strategies, we haven't designed one yet because we are planning to hopefully participate in the next iGEM competition. And in that competition, we would plan we already have a business model, but we need to perfectionate it because we need to consult more stakeholders. But we already talked with the farmers about our solution, and we explained that it was a biopesticide, a simple explanation of how synthetic biology works, and they told us that yes, they would buy it if it is effective. So we think that our target market is interested in our solution, but we haven't designed yet a marketing strategy. But that is our plan for the future.
Awesome. Thank you.
Okay, we are already over the first 10 minutes. Farouk, I saw that you also had a question for the team.
Yeah, actually, I had a question about regarding the human practice part. I saw the programmatic music part. Can you just talk a little bit about it?
Sorry, can you repeat the question, please?
Okay, go Andre.
Yeah, we wanted to make an art project because we have several members in the iGEM team that play music, and programmatic music is a concept that we took from Kandinsky that combines shapes and colors, and it fusions with the musical notes to represent it. It is a concept called synesthesia,
I don't know the word in English, sinus I'm not sure, but yeah, we wanted to represent the, well, fusionate music with science and be able to represent the proteins, the form of a protein in the musical notes. So we chose different chords for different amino acids that conform the peptide. And well, the chords were chose randomly because it is a composition in mostly minor tones because we like minor tones. And yeah, that's why we came up with that idea. We wanted to represent the protein in music.
Great, thank you so much.
Please go, Adrian. Do you have a follow-up answer?
Yes, I would just like to add one thing, that one part that I pretty much liked about the ARCS project that our teammates did is that the composition is divided into 3 different phases. Each phases represent one stage of the complexity of the protein, like the linear amino acid sequence, the beta sheets structures, and the third movement is the oral structure of the protein. So as the structure and the complexity of the biological parts grow, it also grows the complexity of the pieces. So that's one thing I like very much.
Thank you so much.
That sounds really great. Please go, Beatriz.
Your project uses a very novel application of this spider peptides. And as you mentioned, they're natural uses to be injected into the prey. Do you have any idea how stable it would be in the plant itself and until it gets ingested by the bug? Do you have any stability data or guesses, I guess, on this peptide?
Okay, Adrian.
Sorry. Yes, we know for literature that they are stable at 30 degrees Celsius for about a week. Yet we have a proof of concept that we want to prove how stable can they be in different situations for the next competition.
Okay, I think we still have like more minutes to have more questions. I don't know. Yes, go please, Rudy.
So follow up to the question that Beatriz posted. If you were to modify this peptide to infer characteristics that are important for this project, how would you go about doing that?
Pardon me, could you please repeat the question, please?
Yeah, so Beatriz was asking about she was asking about the stability of this peptide in different conditions, right? So let's say if you were interested in optimizing the pH condition, besides making this peptides that are joined together or fused, how would you go about improving even further either a pH characteristic or a thermostable characteristic? So you're saying 30 degrees centigrade for a week. If you want to improve that, how would you go about that?
Yes, Manuel.
I think one option to improve the stability of our peptides is to work in the final formula of our product to look for some, I missed the word, but substances or solutions that help to the stability of the peptides. And this is it will be the way that we will present our final product, but we need to run some essays to prove the stability of the peptides.
Okay, thank you. Yeah, and what you just described will be like a formulation improvement. Thank you.
Okay, thank you for your answers. I think we have time for a last question or last two questions. Yes, Rudy, did you have your hand? No, okay, please go, Beatriz.
You briefly mentioned that already, but how do you envision the final product being applied to the plants? Is it a spray solution?
Yes, from our engagement with the local producers, we ask for like these properties these products should have for them to be able to use it. And one thing in common they all said is they would prefer a solution, a liquid solution that would be sprayed into the fields. So that would be pretty much how we would apply it.
Okay, just a final thought, please, Gracie.
Yes, just to complement that answer, the farmers already use hazardous agrochemicals and they spray it. Well, they spray them. So we don't really want to change the way they use them. We only want to implement our solution, but in a natural and organic way, so they can use it the best way they can. Okay, thank you so much.
Okay, thank you, team. UN team, that's all for today. Thank you for your questions. Thank you for your answers, and you can leave the session now.
Congratulations, team.
Thank you.
Thank you.
Thank you.
Thank
Bye.
you.
Very much. Goodbye. Have a nice day.
Okay, with this now we are welcoming the team AgroBio. So please remember that I need your mics off, your camera on, and the way this is going to work, please raise your hand. The judges are going to ask you a question and you can answer opening your mic, and also if you have like following up answers or complementary information, also you can go with it. So are you ready? So we can start? Yes, no, maybe?
Yeah, we are. It's lovely to be here. Thank you.
Okay, can you tell me if all your team is already here or are we waiting for someone else?
We are ready to go.
Okay then, so welcome. And who wants to start with the first question, judges? Okay, please go, Beatriz.
I can start again. So in your project, you want to increase the amount of soluble phosphorus in soil. Let's assume that your chassis works great, your constructs work great. What is the effect of excess phosphorus in the soil?
I'm sorry, what is the effect of that excess of phosphorus in the soil?
Yeah.
All right.
Yes.
Yeah. So our goal is to standardize and to carry out such protocols that there is no excess in the soil. So our protocols include designing such a circuit and such such a circuit that in vitro in the laboratory, where it's going to take place all of our project actually, it's just the right amount, so it's not an excess of phosphorus. We attached protocols to first. Maybe someone else can follow up regarding that in a moment. So it doesn't happen in real life.
Okay, if we don't have like more additional comments, Rudy, you can ask your question, please.
Yeah, so very interesting project. When it comes to the enzymes that you're expressing, I assume that you want these enzymes to be expressed extracellularly, and the reason for that is because the phytic acid that is mentioned is in the soil, right? So when you're making your construct and you wanted to make sure that this enzyme is expressed extracellularly, what would you do to make sure that that happens with your construct?
So we found certain evidence that although not all of the phytase, which is the one enzyme that catalyzes the reaction from phytic acid to inorganic phosphorus, although not all of the enzyme manages to go beyond the cellular membrane, we did find some evidence that it does a little bit. So part of the improvements we have already noticed was that we could have included and considered mechanisms. So we are sure most of the manages to travel right from the intracellular environment to the extracellular matrix.
Okay,
thank you.
Please, Sikak, ask your question.
Okay, thank you very much. I want to commend the team for a very splendid work. When I was going through their project, I was impressed about the work they are doing on the use of phosphorus solubilization organisms as an alternative for conventional fertilizer in order to improve bioavailability of phosphorus for sugarcane. I just have one quick question for you. How would your project run for multiple years? In other words, how do you intend to sustain your projects?
So we're working in partnership with Ingenio Magdalena. So our main goal is to not stay in this phase of competitions, but to express our project in other microorganisms, like microorganisms from the soil. So our main objective here is not to just stay in the competition, but to make it a reality, work with local farmers and such. So the part of the community that lives there and uses culture as a means for income and food would be our main focus there. We will also create a biofertilizer. So basically, we wouldn't stay in the starting phase. We would like to continue, even though our competition might be over and our participation in iGEM might be over. Because we have the partnership with Ingenio Magdalena, the project would continue.
May I just briefly add? May I just briefly add that Ingenio Magdalena, the sugarcane plant that sponsored us, they are genuinely interested in switching to a business model based on biotechnology. So they are absolutely invested, and we propose, as you may know, here trying in vitro with E. coli, and then moving on to different stages where we express the system in native microorganisms to be isolated from the sugarcane plant soil.
That's great. Thank you.
Okay, Jasper Brook, please go with your question.
What makes your project is better alternative than conventional fertilizers?
And our project would be based from a bacteria. So instead of using normal fertilizers that exceed the amount of phosphorus that is needed by the soil, and because this phosphorus passes into the phreatic, passes to the natural water resources and to the soil, it makes no compensation. So it would be contaminating. Instead, our project would be use less materials for like a way of saying, and since it's a natural form of regulation, our bacteria will be producing, but in a sustainable and responsible way to avoid contamination.
Thank you.
Okay, please go, Beatriz.
Your project involves applying the modified chassis into the soil, right? Have you given any thought on biocontainment of this modified organism or how it could affect the microbiome it's in?
Yeah. First of all, we proposed our vector to be expressed in E. coli, but this is just the first step because we don't want to introduce E. coli in the natural microbiome. This is just to propose an initial vector. Then we need to optimize and find another expression system based in a native microorganism. So if we introduce that bacteria, it won't affect significatively the microorganism population.
And may I just briefly add, though not considered in this project because of the reason my fellow exposed, we did consider in further stages biolabeling, so we can know exactly what strain specifically among several different strains we're going to be tested are we using right now, though maybe that's not a measure when it comes to biocontainment, but at least for bio identification. So yeah.
Okay, thank you for your answers. Does the judges have another question related to their project? Okay, please go, Beatriz.
I can go again. I'll follow up a bit on what Rudy asked about signaling and intracellular versus extracellular environment. How would inorganic phosphorus be transported inside the cell if your enzyme is being expressed intracellularly?
At least for E. coli, which is at the stage we are right now, E. coli has native pore proteins that allow most forms of phosphorus to travel between cells, this intracellular environment and the extracellular matrix. But that is most definitely one of the things to be considered when switching from one expression system to another, most definitely.
Okay, well, I have a question. How did you approach all your education perspective, considering like your whole project?
Well, at the beginning, we started with conferences at the biochemistry, microbiology, and biotechnology congress at Universidad del Valle de Guatemala. So our team advisors, they gave conference about what is genetic engineering, what is about solubilization of phosphorus, the use of microorganisms in the aid of biofertilizers. And we also hosted a radio talk show in 949, which is one of the main radios that the youth population connect to, I guess. So we were talking about what is the competition, why are we participating, what were our main objectives, and basically the use of genetic engineering in the aid of agricultural processes.
Okay, thank you. That's a great approach. And Rudy, do you have a follow-up question?
Yeah, just a question about perception. So I think E. coli typically has a bad rep because there's a lot of E. coli contamination that results in food safety issues. And I know that a lot of E. coli strains are not pathogenic. There's only a select few that are. But when you propose E. coli to be used in this project, what kind of perceptions do you get from the potential customer of this, and that you're actually adding something that can sound bad into something that can be used for food?
Then again, so you can have a little bit more context. There is another research group that is working simultaneously right now in the bioprospection phase with the plant. So what we were proposing here was just at the same time they find the most suitable microorganism to use. We're proposing using just E. coli as a proof of concept, and we are ready whenever we already have that that native microorganism to use. So there is the intention would never be to release E. coli to the Ingenious soil. Just, if anything, a few very controlled assays in retired lands from the Ingenio.
Thank you.
Okay, then I think we only have a few minutes left. Do you have any other questions? Yes, Beatriz?
I have an application question. Does the Ingenio measure amount of phosphorus in the soil, or do they just apply a fixed amount of fertilizer? And I'm asking because let's assume in the future your system works greatly. How would they know how much fertilizer to add? So sort of to overkill in phosphorus and actually reduce the amount that they're applying.
Do you have additional information about this part?
Sebas, would you like to add something in that regard?
Right now, I don't have the exact number, but I remember that they have a goal of to reduce the use of fertilizer, phosphorus base, in 33%. So the goal will be to, with our system, achieve that goal of solubilization.
Okay, thank you. Rudy, do you have a, sorry, Beatriz, do you have a follow-up question? Okay.
No, I was just saying thank you.
I'm sorry. Okay, well, I have a last question if any of the judges have more additional thoughts about it. I was wondering how well accepted your project is considered in your country. Did you have like any challenges related to the production or your main project by itself?
Yeah, mainly, I think Guatemala is in a stage that little is known about biotechnology, and not only by the scientists that have been working in this area, but mostly about the general public. So it's about mostly going further and trying to look about how to overcome all the problems that are with genetic engineering, because there is a lot of disinformation, ignoration regarding this topic. So we would first need to develop some certain education tools that would allow us to get to know